My name is Joseph, and today I'm going to talk to you about a number of roles that oxytocin neurons have within mammals. If you haven't already, please fill out the survey situated on the homepage. Oxytocin has been represented in pop culture as a kind of love hormone responsible for enhancing romance and having relationship improving effects. These roles have been heavily exaggerated, but however do hold some level of integrity behind them. This resource shall provide you with an in-depth overview of its true functions and some of the research that led to these discoveries. As of recent times, the majority of study into oxytocin focused largely on its involvement within the social behavior connected to maternal attachments and pair bonding. However, there has been less research into its role in social interactions, not in the context of reproduction. This review will delve into these behaviors. Oxytocin is an ancient neuropeptide hormone consisting of a chain of 9 amino acids. It activates the oxytocin receptor via its release from the pituitary gland. This sequence of peptides has been conserved in vertebrates during the course of evolution. The majority of oxytocin production comes from the supraoptic and the paraventricular nuclei of the hypothalamus that can be seen in the figure below. Oxytocin can be released both peripherally by the posterior pituitary gland and centrally via direct neural release. The peripheral release of oxytocin is in control of physical reproductive functions. Its effects of muscle contraction are required for processes such as the induction of labor and lactation. Oxytocin controls lactation through its coordination of the let-down reflex. It binds to oxytocin receptors that are present in mammary epithelial cells and accelerates casein intracellular transport, resulting in myoepithelial cell contraction. These reproductive roles that oxytocin have may be accountable for it being highly conserved in vertebrate taxa. The central release of oxytocin is thought to have a much wider effect on complex social behaviors. To be social within one species can involve a range of behaviors that are not comparable with other mammalian species. For example, manners of aggression, grooming, and pair bonding differ greatly across species. This section of the resource will cover the most extensively researched functions of oxytocin, including maternal affiliation, pair bonding, and its function within family groups. Many studies have identified that oxytocin is a key hormone in the facilitation of maternal behavior. For example, Peterson and Prange demonstrated that the administration of 0.4 microgram of oxytocin in naive virgin female rats would cause them to show full maternal behavior towards infant rats. To reinforce oxytocin's connection to maternal affiliation, a number of oxytocin knockout mice studies occurred. They observed the loss of function mice and monitored the mutation-associated behavior. Oxytocin knockout mice displayed a loss of maternal behavior when compared to wild-type mice, thus highlighting oxytocin's significance as a hormone responsible for maternal attachments. Another role that oxytocin neurons have is within recognition in the early social environment. A comprehensive cross-disciplinary search of studies by Skatliff et al. Identified a correlation between higher oxytocin levels and parent-infant contact, indicating that oxytocin is a pivotal hormone in social recognition. This correlation is confirmed through the use of intracerebroventricular receptor antagonists. Nelson and Pansep inhibited scent recognition between 15-day-old rat pups and their mothers by blocking the activation of the oxytocin receptor, reinforcing oxytocin's clear position in social recognition. Oxytocin has a role in a variety of social behaviors, and is theorized to be largely responsible for the mechanisms underlying monogamous behavior and selective pair bonding. Animal studies have demonstrated that oxytocin receptor signaling drives pair bonding actions via the formation of strong relationships between monogamous partners. Monogamy in mammals is relatively rare, only occurring in a total of 5% of species, and our understanding of oxytocin's importance comes largely from studies in prairie voles. The monogamous prairie vole forms close relationships with its mate following frequent acts of copulation. Through the use of oxytocin antagonists, Insel et al. Identified that pair bonding could be prevented without the alteration of sexual behavior. Additionally, they discovered that central oxytocin injection allowed pair bonding to happen between voles even in the absence of copulation, providing subsequent evidence for an oxytocin pair bonding correspondence. Several oxytocin receptor studies on similar non-monogamous species, such as the montane vole, were conducted to support this oxytocin pair bonding relationship. A quantitative comparative study by Wang et al. Revealed that many brain regions, such as the nucleus accumbens, did not exhibit coherent differences in the patterns of oxytocin receptor expression between the two species. According to this study by Wang et al., there are few consistent differences in the patterns of oxytocin receptor expression between the two species. They revealed that very little difference in receptor expression in brain regions such as the nucleus accumbens. This led to research to believe that the differences displayed in social behavior and pair bonding are likely due to differences in oxytocin receptor distribution between the monogamous and non-monogamous species. Many studies employing non-human primates have been conducted as a result of the challenges in adapting oxytocin studies in rodents to people. Using marmosets, Smith et al. Investigated the function of oxytocin in heterosexual primate relationships. Using oxytocin injection and oxytocin receptor antagonists, they assessed its impact on partner preference and sociosexual behavior. This study demonstrated that modifications in oxytocin activity affected the formation of pair bonds in marmoset monkeys. Furthering this, Grebe et al. Carried out a comparison study of the oxytocin neurocircuitry in monogamous and non-monogamous monkeys found in Ulama. The mapping of oxytocin receptors in the 7 different species of lemurs, in contrast to other studies carried out in rodents, found minimal evidence for neuropeptide circuitry particular to this type of pair bonding. The similarities visualized in oxytocin expression suggested that there are evolutionary distinct mechanisms for monogamy and pair bonding.